afternoon. It's a great pleasure to be here. I am Guillaume Filion here from Barcelona. And this here has got nothing to do with my work. It's a piece of art by Shigeo Fukuda, who is a shadow artist. And that's to remind myself when I thought that what we see depends on where you put the light. So this artist likes to play with the shadows so you can see the face of the guy appearing just from letters. So what we do is actually chromatin. We're interested in chromatin, and we do this in the model system of Drosophila. And most of you think of Drosophila like this, but I'll be speaking of Drosophila in this form here. So everything that I'm going to show is in Drosophila, but in Drosophila cells. So this is the system that we work with. It's going to be clear by the middle of the talk why we need to do this in cells and this cannot be done in full organism. So my lab is interested in chromatin, in particular in chromatin from a top-down view instead of a bottom-up view. And a couple of years ago, when we started to be interested in chromatin, we were starting with a very simple question, which is how diverse is chromatin? How many types of binding patterns can we identify in chromatin? And to answer that question, we mapped 53 chromatin proteins in Drosophila cell line. And you can see the chromatin proteins here. I don't expect you to be able to read the name. It doesn't matter. What matters is the amount of them that there is. And this here is not a ChIP-seq track, it's a DAM-ID track. So if you don't know what DAM-ID is, it doesn't matter, it's just like ChIP-seq. What you're seeing here is a genome track, so that's the coordinates here with the genes and the real position in KB. And when you see a little mountain here, it means that a particular protein is present at that spot more than others. So that quantifies the amount of time that a protein spends on one particular spot, or the amount of protein that there is on that spot in the cell population. So I've arranged the profiles in this slide so as to highlight a little bit the order that you can see already when you do something as simple as this. It's particularly obvious that there is clusters that appear. So for instance, this one's fairly visible. There's 4 proteins at the bottom that seem to have pretty much the same profile. You can see this again up there. For those of you who are at the talk of Jen Ernst, then this is because there is so much redundancy in the chromatin that actually you don't need to have them all. At some point, things start to look the same. And so how many types can we find in this in the Drosophila chromatin? Well, there's plenty of ways to answer the question, but the way we answered it is to do a principal component analysis of this data here. So this is how the projection looks like. Each dot here is one probe on the microarray. This was done on microarrays. And 2 dots are close together in this projection, not when they're close in the linear genome, but when they have similar profiles of proteins that bind to them, or chromatin features. And we can see here, so with the help of the color, that there is 5 domains. So it's not very visible in this one. When it rotates in 3D, it's much more visible. In particular, the green cloud that goes in the background here is invisible. But there's 5 lobes that come out of the cloud, which means that you could say there is 5 signatures of chromatin in the Drosophila genome. Because if you're in one of them, then you're very close to all the other ones, which means that the signature would be relatively uniform. But if you're in black, you cannot be in yellow, which is another signature. So now when we map them back on the genome, we obtain something like this. So that's the same map as I've showed to you before, but this time I've highlighted what the classification methods tell you, and you can see that we recover what we had identified. So this here turns out to be the blue color. This here turns out to be the green color, but we also recover a little bit more that these big blocks that we saw in the middle here that looked uniformly for the human eye are not uniform at all for the computer. It seems that there is 2 chromatin types here that are in yellow. Both of them are active, highly transcribed, but they're very different from the perspective of what proteins bind there. So that was work that was published a couple of years ago, and one of the biggest surprise that was associated with this work was what we call black chromatin. So black chromatin, I'll show it back here. This is a typical locus, nothing special here. Black chromatin covers about 50% of the Drosophila genome, and as you can see, it's characterized by having almost nothing. So that was a little bit of a surprise because we were claiming that about 50% of the Drosophila genome doesn't really see transcription factors. They're just not binding there. The other surprise is that 66% of the genes that lie in black are not expressed, they're silent. And the converse is also true, that most genes that are active are in black. So it was not histone marks that were the best predictor for gene expression. It was just the absence of everything. That was really the best predictor for not being active. So we thought, okay, maybe we have something great here. Maybe we have discovered a new chromatin type that's repressing transcription. And this chromatin type would be characterized by having nothing. So that's the question that we've been working on for a couple of years now. Does black chromatin repress transcription, or is this something completely unrelated, or more likely something that's downstream of the live transcription? And now we have a problem that we cannot really modify the system. Here, we cannot just transform black chromatin into red chromatin. So that would be the experiment we want to do. We have this magic machine that would transform black chromatin into something else, and then we see whether the genes would go up or not. But we cannot do that experiment because we cannot modify the system. Chromatin cannot be modified. But we're the first people to have a problem of that sort. So many people who work in science in general have a problem that they cannot modify the system. And in particular, people who do climate research, or in particular, research on the water currents, cannot modify the system. The system is the Earth, so they cannot do the experiment they want. In particular, if they're interested in water currents, where does the water go, etc., it's not trivial because they can test their models. And then what do? Well, they wait for the fantastic experiment for luck. In 1996, a ship that was on its way from China to the US sank exactly there. And on its board was a cargo of tens of thousands of rubber ducks. So here was a release of tens of thousands of rubber ducks, and then what happened? What happened is what had to happen. The duck started to drift, basically. And from between 96 and 2010, more or less, they have actually gone around the world. And this was the perfect experiment because it was for free, they were completely neutral. And when you have this army of ducks that were coming along the coast, then of course people were freaking out. They were calling the Guard Coast, saying, we've got this army of ducks, what do we do, etc. So it was even free for the scientists to track the ducks because people would report them all the place they went. And so in a couple of years, in 1 year, they went to Alaska. And in the end, they were the unproof to that day that there is a water current that in 10 years goes from here to here on the other side of the America. And then after this, they went down here and crossed even the Atlantic Ocean to go to Europe. So the bottom line is that you cannot modify your system, you can still saturate it with reporters. And so the main idea of what we do is we do the reverse experiment. Instead of changing the chromatin, we bring the genes to a different chromatin type. So we want to take a gene that's active, this one, put it in black chromatin, and then see what's happening. Is it going to be inactive or not? As simple as that. So that's not the duck approach. The duck approach is this one, is to compensate quality by quantity. If you do that so much that you're going to have a lot of reporters, then you can get a good quality picture of what's happening in the chromatin. So that's the experiment we want to do. We want to take one gene and put it on many, many, many locations. And the way we do this is by tagging every gene here with a barcode. So we need to be able to distinguish the different copies of the gene. Otherwise, they'd produce exactly the same transcript, and it would be impossible to distinguish one from the other. So the transcriptional output of all these guys would be indistinguishable. So this here is called thousands of reporters integrated in parallel, so we call it TRIP. And I'm going to tell you a little bit more of the experimental detail how that works. So again, everything is done in this cell line that I've showed at the beginning. We have a transposon here. That's a plasmid on which we clone different promoters that are here. And here the barcode is 20 nucleotides that are completely random that are put in the plasmid in such a way that every copy of the plasmid has a different barcode. So these ones, for different molecules, they're different. And we put the GFP here in the middle just to be able to see the transcriptional output. So the barcode is in the RNA, but not in the protein. Okay, it's after the stop of the protein, so it doesn't influence the protein, but it's in the RNA, so it will be expressed together with the RNA of the GFP. And the transposon here can be co-electroporated with the transposase, and the transposase gets this, and then puts the transposon somewhere in the genome. So this here is the duck approach. We can do this with high throughput, shotgun approach, taking the same exact sequence, except the barcode, and put this in different contexts and see how it's going to behave. When it's integrated, it looks like this. So that's the transposon here. It produces transcripts like this, and you can see that all these parts here of the plasmid have been kicked out during the transposition. Typical experiments look like this. So we do an electroporation in these cells. At day 2, 80% of the cells are GFP positive. We take with transposase or without transposase, which are control. And as you can see, without transposase, within 15 days, we go below 10 and then 1%. So there's a bit of residual insertions that stick somewhere in the genome. But with transposase, this is much higher. So we can have about 35% of the cells in which the integration has been integrated, the report has been integrated in the genome and stays there, and it's going to stay there forever. So Now, how do we measure expression? So we use the barcode. We don't really use the GFP. The GFP is just for the experiment. And the way this works is fairly simple, actually. So we transcribe here from the promoter that's here, and we see the barcode here in the RNA and the DNA. So we make RT-PCR and a PCR with the same primers, and we take the ratio RNA divided by DNA, and that gives us the expression of that particular barcode in the population. That's how we can know how much one particular barcode is expressed, and because we're able to map the barcodes, we can associate one particular location to one particular expression level. So the first thing that we notice when we do that is that the position matters, but this was kind of expected, at least for people who were studying chromatin and heterochromatin, this was expected. So we have 6 promoters that are highlighted here, and this is a no promoter control. On these valence plots, the left one represents all the insertions that are on the chromosome arms, and the right valence represents the insertions that are on the centromeres. And you can see that all the promoters are sensitive to being inserted in centromere, so the expression goes low. So this one, number 1, particularly sensitive to that. When you put it in centromere, it gives up, it doesn't transcribe anymore. And you can see that all of them are quite sensitive to that. So that's the first news, is that transcription depends on the context. But that we knew, that's a sanity check, but depends on the context a lot. Does it depend on other contexts? So the summary is all promoters are sensitive to heterochromatin. Another context that's known to affect transcription is called lamina-associated domains. So here they represent where the locus is in the genome. So when you have a piece of DNA that touches the lamina, you can detect it again by DMD, and this is the signal that you see here. So that shows how much the locus touches the lamina. So when it's high, it means that the locus is against the lamina. When it's low, it means that the locus is inside the nucleus. That's one of the promoters here. You can see that there is a very good visual anticorrelation between the expression of the reporter and the expression, the position of the locus in the nucleus. Okay, it's especially clear in that regions here, that when the locus touches the lamina, the expression is low. So here, these are the valence. The left one represents the one locus is inside the nucleus, and the right one represents when it's at the lamina here. You can see that the first 2 promoters are not really sensitive to lamin, but the next 4 are quite sensitive to lamin. So whatever happens at the lamin, whatever creates that here, is not the same as what happens at the pericentric heterochromatin, because only some kinds of promoters are sensitive to lamin and some others are resistant. So these are 2 different pathways of inactivation, of epigenetic, if you want to call it activation. So an interesting case that actually bugged us for quite some time is the BUN gene. So the BUN gene here is one out of many cases that we've observed. It's a long gene. It's 100 kb, which is very long for Drosophila, and it's at that position here. And you can see the insertions are not expressed when they're in the BUN. Okay, so all the insertions that are there inside the BUN gene are down. They're not really expressed. And yet the BUN gene is expressed itself. It's highly expressed. You can see that it's not in LADs. So these boxes here represent the LADs. So it should be, the insertions should be expressed according to the previous criterion. It's full of enhancers. So these are development enhancers and housekeeping enhancers. You can see that it's even enriched in the BUN gene, and yet these enhancers are not able to activate the reports. So, and I forgot to mention, it's full of transcription factor that bind there. It's pure euchromatin. You can call that euchromatin. So why would it be the case that insertions in euchromatin are not able to be expressed? So we were looking for this for a long time, and in the end, the answer turned out to be quite surprising for us. So why insertions in the BUN gene silent? And I repeat that this is one of many cases that happen genome-wide. So I'll remind you a little bit about the HICE experiments. Of course, then I'm kind of spoiling about the answer. The HICE experiment allows you to see when 2 sites are in contact in the cell. The way it works is you cross-link the DNA like this, you cut it with a restriction enzyme, and then you ligate. So when these pieces come together, you can detect it by throughput sequencing, and that tells you that there's a contact between that and that chromosome. So we can see the HICE contacts usually with contact maps that look like that. So the BUN gene is here. The LADs are marked again. And if you look carefully at the pattern you see here, it may be apparent to you that if you look at the diagonal like that, okay, there is some puffs, and there is some types where it's thin and sharp. And right in the middle of the BUN gene, it's here, okay, and it's thin and sharp. And in the LADs that are next to it, like here, for example, this is a LAD, as you can see here, this is also thin and sharp. And so the best predictor that we have found so far for the gene expression is not whether something is in LAD or outside the LAD, but it's whether this thing here is thin and sharp, or whether it's blurry and fat like that. Okay, and so the promoter of bun that is here is actually here in a puff that's like there. So it really looks like, in reality, what matters for transcription of the reporters is not whether they are the lamina, whether the chromatin in terms of high C looks like this or looks like that. What does that mean? Not quite sure. So we can do the reverse and then focus on all the active insertions for the inactive insertions. And this time we look at how much the contacts decay with the distance here, and that's almost a perfect linear trend in log-log, so that's a parallel decay. So when the reporters are active, the decays look like this. When the reporters are inactive, the decay look like that. So it really seems that in the end, it's more about how the locus is folded than whether it's at the lamina or not to predict genome expression. So does black chromatin press transcription? The answer is then yes and no. In reality, no, because when we take that into account, then all the predictive power of black becomes 0. But because black associates with these features, then it looks like if you do the test naively, then you'd say yes, it represses transcription, but when we take these effects into account, it doesn't anymore. So this is not a new mechanism for repression. The real mechanism seems to be the genome conformation. So heterochromatin and LADs do different things. There are two different pathways of gene silencing. The 3D structure explains LAD position effects, probably not about being at lamina or not, but more how the locus is compressed or is it folded. And also the kind of sad news for all of us is that predicting gene expression from sequence will be hard, because here in all these examples, the sequence hasn't changed. Okay, so it's just the locus that has changed, and that explains for a factor about 10 of the gene expression level, which is a big factor. With that, I'd like to acknowledge my team people who did the work. So this is mostly the work of Marc and Arantxa, but everybody else is contributing to the TRIP method in the lab. I'm very happy to take questions. Thank you very much.
We have questions from Guillaume from the audience.
Thanks for the very nice talk. Did you actually also correlate to the number of annotated enhancers?
Yeah. So that's a little bit what we're seeing here. Okay, these are star-seeking answers, if you know the method. In general, the correlation is good. But the problem with this experiment is not that the correlation will be bad, because it's always good. It always correlates a lot with something. And it's very difficult to disentangle which are direct correlations and the indirect correlation. So as you can see here, the enhancers are enriched in bun here, and yet all the insertions are down. So it looks like the conformation or something else in the bun gene trumps the enhancers, local enhancers.
Enhancers which are specific for this cell type, or
yeah, they were done in this cell type. So they're active in this cell type, but maybe not on the promoters that we've chosen. Okay, if there is some kind of combinatorial code between enhancers and promoters, then we'd miss it, for instance.
Okay. Yeah, I can ask one more. If you correlate to the description of the nearest genes after the transcription, is the chance actually where you integrate the sites higher if you have more transcription in the area or not?
Yeah, there's again a very good correlation between the expression of the transgenes and the expression of the local endogenous genes. One of the best predictors, except the 3D structure, is how far is the closest expressed gene.
And the integration? So do you also integrate, because it's not uniform, right? So it's randomly integrated.
Yes.
So therefore, where you integrate, is that more likely at areas which are transcribed?
A little bit more, yes. Yes, it's a little bit more in exons, actually.
Thanks.
I think we need to move on. Thank you, Guillaume, again.